Dear friends, greetings from Ishita Ray, Assistant Professor, Department of Zoology, Shoshona College, University of Calcutta. Today we are going to discuss about a very important molecule of our adaptive immune system that is known as major histocompatibility complex, or in short we call it as MHC. Now, MHC is a tightly linked cluster of genes located in the HLA locus of human chromosome number 6. The product of these MHC genes can be classified into two categories, class 1 MHC and class 2 MHC. As we see in the picture, on the left hand side we have the class 1 MHC molecule and on the right hand side we have the class 2 MHC molecule. Now, what is the function of these MHC molecules? MHC molecules perform the most important function by detecting or by distinguishing between a self molecule and a non-self molecule. That is, for example, a foreign molecule when enters our body, that is considered to be a foreign or non-self molecule by MHC molecules. Now, class 1 MHC is present on the cell surface of any nucleated cells, whereas class 2 MHC is restricted on the surface of APC, that is antigen presenting cells, and that of B cells. So, whatever be their presence, their main function is to distinguish a self antigen from a non-self antigen. Now, let us look into the structure of the MHC molecules. The class 1 MHC is made up of a polypeptide chain which is called the alpha polypeptide, along with a short polypeptide which is called the beta 2 microglobulin. Now, this alpha polypeptide has been folded, or it shows the presence of three folds which are called alpha 1, alpha 2 and alpha 3. These folds are called domains. So, we can see in the diagram that the membrane distal domains of class 1 molecule, that is the domains which are located away from the membrane, they are classified as alpha 1 and alpha 2, whereas the membrane proximal domain is the alpha 3 domain. Along with this alpha chain, we have another very important constituent of the MHC class 1, that is called beta 2 microglobulin. Now, the association of beta 2 microglobulin with the alpha chain is extremely important. If this association does not happen, the MHC will not be able to perform its function. Now, we see in presence, in between alpha 1 and alpha 2, we find a molecule, we find a structure which is called the peptide binding cleft. Now, this is the structure, or this is the place where the MHC binds with the antigen and presents the antigen to the T cell. So, as we see, the class 1 MHC has got three parts as a whole which are lying outside the membrane. One portion which is called the transmembrane segment, and another which is present towards the cytosolic side of the membrane which is called the cytosolic tail. The transmembrane segment of the class 1 MHC remains embedded within the plasma membrane of that respective cell, whereas the cytoplasmic tail remains projected towards the cytoplasm. Okay, so let us move on to the class 2 MHC molecule. The class 2 MHC molecule is present on the surface of any antigen presenting cell and that of B cell. So, class 2 MHC molecule has been divided, or it is made up of two peptides. One is the alpha chain, another one is the beta chain. So, please, we can actually differentiate between the structures of the two MHC, class 1 and class 2, by looking at the diagram simultaneously. In the class 1 MHC, we were seeing that there was only one alpha chain and one beta 2 microglobulin, whereas in case of class 2 MHC, we are seeing that there are two polypeptide chains, alpha chain and beta chain. The membrane proximal domains of the class 2 molecules are alpha 2 and beta 2, that is, these domains are located closer to the membranes of the APC or the B cell, whereas the membrane distal domains are the alpha 1 and beta 1. and the peptide binding cleft, that is the portion where the antigen will go and bind with the MHC, that is located in between the alpha 1 and the beta 1 domains. And it similar to class 1 MHC, class 2 MHC also has similar transmembrane segment by which it remains anchored to the membrane of the APC or B cell, and it is also having a cytoplasmic tail. Now, this is, I mean, in short, a brief description of the structures of class 1 MHC and class 2 MHC. Now, let us see that class 1 MHC, which T cells are being presented by these respective MHC molecules. So, class 1 molecule actually presents antigen to cytotoxic T cell, and class 2 MHC presents antigen to TH cell. So, class 1 MHC is responsible for TC cell activation and class 2 MHC is responsible for TH cell activation. Whatever be the type of T cell, the molecule of T cell which interacts with the MHC, that is referred to as the TCR. That is, the TCR or T cell receptor will interact with the peptide binding cleft or the membrane distal domains of both the MHCs. So, friends, it is quite evident that without the presence of these MHC molecules, it is not possible for the T cell to interact with an antigen. So, that is why the MHC plays a vital role in T cell activation by presenting the antigen to the TCR. So, that is it. Thank you, friends.